Hello, I'm Jacob Evarts.
And my name is Mikala Capage. Our mentor is Dr. David Garcia, and today we're presenting our poster, Hunting for Prions, Propagating Putative Prion States in Budding Yeast.
It is well known that stress conditions can negatively affect how yeast cells grow and propagate. Garcia Lab is working to illuminate the mechanisms that are employed by cells to overcome these challenges. In particular, epigenetic mechanisms, or systems that are not a function of the cell's DNA, have been of particular interest. Prion proteins were the epigenetic mechanism that we studied in our experiments. Prions are proteins that have an alternate conformation compared to their normal or naive shape. Despite being the same amino acid sequence, their prionic forms can have different behaviors than their naive forms due to this shape change.
In this project, we asked if 6 strains of yeast that have exhibited resistance to the effects of harmful chemical stressors pass down those traits in patterns consistent with prion proteins. The 6 strains are known in this experiment by the enzymes we suspect to be in a prion conformation. These enzymes are ABD1, SET1, ppm2, PUS4, PUS6, and TRM5.
The protocol we used in the experiments involves 3 steps for screening for strains of yeast that are potentially prion harboring. First, we propagate and isolate the candidate strains. Next, we perform growth assays on these strains, usually under stress. And finally, we analyze the resulting growth dynamics against control strains that do not have prions.
Prions and the traits they cause inherit in unique patterns distinct from traits caused by genetic mutations. Namely, prions always inherit into a diploid cell and, through meiosis, in 4:0 or 3:1 inheritance patterns. This is known as non-Mendelian inheritance.
These unique inheritance patterns are due to the fact that prions are proteins that exist in the cytoplasm, generally, and are thus considered cytoplasmic elements. The unique patterns make it easy to screen for the presence of prions by testing the diploid dominance and inheritance. We found that 5 of the 6 candidate proteins had strains that were dominant in a diploid background, meaning the diploids always inherited the phenotype. Additionally, the same 5 strains showed 4:0 or 3:1 inheritance in their progeny, whereas a 2:2 inheritance would suggest that the phenotype was genetic in nature. Only ppm2 did not exhibit these phenotypes, suggesting that its growth dynamics are not prionic in nature.
This project set out to determine if 6 strains of yeast suspected of harboring prion proteins did indeed contain prions. To do this, we isolated and tested strains of yeast. We found that 5 of the 6 strains in this experiment exhibited inheritance patterns consistent with prion proteins. The next step of this project is to test if these strains exhibit other hallmarks of prion-based traits.
We would like to thank Dr. Garcia for his support and mentorship, as well as other members of the Garcia Lab, and appreciate the funding by VPRI Summer Fellowship Program, the Eunice Kennedy Shriver National Institute of Child Health and Human Development, the National Institute of Health, and the Knight Campus.